Hi, I am a student from Harmony School of Advancement, and in this video I will be explaining the IND-21 colony PCR test on a sample of E. coli. In this experiment, I will carry out colony PCR starting with a culture of E. coli that carries an ampicillin-resistance gene on plasmid PUC18. I will first streak the cells over a nutrient agar plate in order to produce single E. coli colonies. These colonies are then used directly as templates in a PCR reaction in order to amplify a segment of the 16S ribosomal RNA genes, which is located on the E. coli chromosome, and a segment of the ampicillin-resistance gene on the plasmid. The materials I used for the PCR mix to conduct the colony PCR experiment include E. coli PUC18, rDNA primers, Taq buffer, Taq polymerase, nucleotides, electrophoresis sample buffer, and PCR tubes. To conduct a colony PCR test, first touch a sterile transfer loop to the solution of E. coli. Then streak the bacteria adhering to the loop on a segment of a plate containing nutrient agar. Then sterilize the platinum loop or use a new polyester loop to make a secondary streak. Repeat these steps 2 to 4 times until the entire plate has been streaked. Then replace the lid on the plate and incubate in an inverted position at 37 degrees Celsius or room temperature. Colonies should appear in about 15 hours at 37 degrees Celsius. Then number 3 0.5 milliliter tubes 1, 2, and 3 on the top with a pen. Place the following into tube 1: 10 microliters of the primers, 10 microliters of the nucleotides, 10 microliters of the PCR buffer, and 10 microliters of the distilled water, as well as 10 microliters of the Taq DNA polymerase. Next, touch the tip of a sterile pipette tip to a single well-isolated E. coli colony on your plate. Then transfer 15 microliters of the solution from tube 1 to tube 2 and add 5 microliters of the electrophoresis sample buffer to tube 2 and place this mixture in the freezer. Then carefully overlay 50 microliters of mineral oil over the liquid in tube 1 and perform PCR for 25 cycles. Retrieve your tube from the tube rack after the last cycle. Remove 15 microliters of the PCR mixture and transfer the mixture to a 0.5 milliliter tube labeled number 3. Then add 5 microliters of the electrophoresis sample buffer to tube 3 and place the tube in the freezer. Then load 15 microliters of the following samples into the sample wells of your agarose gel. Electrophorese until the bromophenyl blue has migrated to within 1 to 2 millimeters of the positive end of the gel. At 170 volts, this should take about 50 minutes. Stain the gels overnight with methylene blue and analyze the results of the gel electrophoresis. After analyzing the agarose gel stained with methylene blue overnight, a stark contrast was observed between the two lanes tested. Tube 2, which did not undergo any PCR, displayed a very low intensity of DNA band appearance, making it challenging to detect DNA migration. In contrast, Tube 3, which underwent colony PCR, exhibited a significantly higher intensity of DNA bands, indicating successful amplification of DNA through PCR. These findings underscore the effectiveness of PCR, particularly colony PCR, in amplifying DNA for detection and analysis. For my English component, I documented my journey into truly understanding the colony PCR test. During the startup phases of the experiment, I set up the basics of my Google site. Afterward, I researched this topic to understand the importance of colony PCR and what the test is trying to accomplish. I then carefully conducted my experiment, making sure that I followed all the instructions while documenting the results on my Google site. Lastly, I analyzed the data I collected and began composing a simple but concise analysis with a conclusion that highlighted all of the important details that I studied. These results were then published on my Google site for others to view and gain their own understanding of colony PCR. For my social studies component, I described the historical background and connections the colony PCR test had on laboratory techniques. The invention of PCR can be attributed to Carey B. Mullis, who developed the method in 1983. Mullis was awarded the Nobel Prize in Chemistry in 1993 for his groundbreaking contribution to science. Carey Mullis's ingenious invention of PCR had a profound and lasting impact on the field of molecular biology, transforming the way scientists study and manipulate DNA. PCR is now a fundamental tool in laboratories worldwide. Thank you for watching Harmony School of Advancement.